So let me give you a case history. This is work that I've done with one of my former interns, actually, who's a superb scientist at the Dana-Farber Cancer Institute, David Rowich. Also is a part-time neonatologist. I call this Finding the Needle in the Haystack. And it's about studying the cerebellum. So I impressed, I hope, upon you on the last lecture just how bad microarrays are, how noisy they are, and how difficult it is to interpret them. So now I want to show you how if you actually do the computation right, you can actually extract useful biomedical information out of it. So the cerebellum is a nice system to study because it has this nicely layered structure. The cerebellum is this part of the brain that sits in the posterior fossa in the back, and it has this very nicely layered structure which is detailed in this cross-section here, so that each layer has a distinct group of cells, and often these group of cells are distinguished by different genes expressed at different times, specifically to that cell type or cell layer. And the cerebellum has a pivotal role in locomotion and posture, but the fact that I can stand approximately straight and move around is almost solely a function of the activity of the cerebellum. And because of its nicely layered structure, the cerebellum has been the subject of a lot of neuroscience because it's much less complex than the other central neural tissues in that regard. So there was a particular system that David was interested in. Sonic hedgehog, it's one of these supposedly amusing names developed by fly biologists, because fly biologists they have a very bad sense of humor, and they apply that to all the genes that they discover. Sonic hedgehog is one such gene. It turns out to be a very important signaling molecule that's very important in proliferation and differentiation of cells. And David wanted to know what was the role of sonic hedgehog in this outermost of the layer of the cerebellum, the external granule layer A, EGLA. It was known that cyclin D1 was expressed in this layer, and that different other genes were expressed in other layers. It was known that sonic hedgehog was important in a variety of different ways. For instance, genes that had previously been implicated in the sonic hedgehog signaling pathway, when mutant, cause in the mouse model here a posterior cerebellar tumor, which is very much akin to the medulloblastomas that are a big cause of juvenile brain cancer, and a lethal one at that. And it's subsequently been shown that medulloblastomas are involved in the sonic hedgehog pathways, a large percentage of them. So the question was, could we find other culprits, other genes that are involved in the sonic hedgehog pathway? And it's a big haystack that we're looking into, because we're looking in two large haystacks, actually. One is a large number of probes on the microarray. Two, we're looking at the entire cerebellum, a complex tissue, even though it's one of the simpler neural tissues, when in fact we're trying to extract a signal out of one part of this complex tissue, this very superficial layer of cells called the EGLA. So can we find the signal in time and space? So we're not only looking for the right number of probes in the right location, but the right point in development. And this was quite a challenge for us, because David is a personal friend and a former intern, and he had heard about, heard me pontificate about the importance of bioinformatics, and here was this close friend who is now engaging in scientific collaboration. And if I gave him the wrong candidates, that means I'd be wasting the time of his postdoc, of which he had only one at that time, and delaying him by 6 months if I gave him some wrong things to look at. So it was a relatively high-stakes game. So the experiment was very simple. To harvest initially just day 5, postnatal day 5 cerebellum, expose it to sonic hedgehog, vehicle, and growth arrest media. Forget about the growth arrest media for a second, but basically exposing it to sonic hedgehog or vehicle, hybridize it to the now obsolete Affymetrix mouse array. The mouse arrays are not obsolete, but this generation is no longer available. And we did what we refer to in our grant as noise models in our bioinformatic analysis, which I will detail shortly. And then we gave a list of 9 significantly upregulated genes and 10 genes not significantly upregulated, and these were going to be subsequently screened with in situ hybridization to see whether in fact these predictions were correct. And so what we did was a very simple thing. If you recall, I showed you in the last lecture that I gave you how noise grew dramatically at lower levels of expression because you could flip the sign of the up or down regulation just by virtue of having low levels of expression affected by low levels of noise. So clearly you need to have some sort of statistical measure of the separation of the two conditions. So in this case, the signal was the expression of a gene in response to the signal, namely sonic hedgehog, and the other is the vehicle-treated ones. And the question is, for each such gene, could we find those genes that were clearly separated across those two conditions? And there's a variety of ways that you could have done it. And in fact, as your problem set, I'll have you explore some of these standard methods that, for instance, are things like a modified t-test, like the significance analysis of microarrays. But I'm going to make the following point, which is trying to understand what you're doing by looking at ratios, and for instance, just looking at a t-test where you're looking at the difference in means is fundamentally misunderstanding the computational challenge in a very simple way. What average do we want to look at, do we want to calculate? And I realize not all of you are computationally involved, so try to keep this simple. There's 2 types of averages that we might consider among many calculating. One is the simple arithmetic average, where you sum the signal and divide it by the number of samples, and you sum the control and divide it by the number of samples, and you take that ratio. Or you take the geometric mean of them. And why would you do one or the other? Well, let me ask you the following thing. This used to, when I first thought about this, I was having the mortgage on my apartment, so it was incredibly important to me. If you have 4 different interest rates over 4 years, such that the amount you're going to have to pay out is 1 plus R1 times 1 plus R2 plus 1 plus R3, 1 plus R4, and you're going to have to pay different amounts on that principal, how am I going to estimate the average interest rate I'm paying per year? Is it the arithmetic average of these R's? No, because there's compounding. I know that's a simple point, but few people realize it's actually the geometric mean. And the same error is being made if you look at the arithmetic mean versus the geometric mean to look at ratios, because fundamentally we're looking at a ratio. And we did the analysis both ways. And I can tell you we knew, in this case, the answer because we did the in situ hybridizations. And first of all, the good news that we published in PNAS a couple years ago was that essentially 80% of the time that we said a gene was expressed in the EGLA based on this simple geometric mean, sure enough, by in situ staining, it was. And when we said that it wasn't, it wasn't. But the point is, if we used the arithmetic mean, we had double the false positive rate. So just understanding something as simple as the difference between an arithmetic mean and geometric mean, which is a non-obvious notion to anybody who's just been used to doing t-tests, screws up the analysis. Let's make this more relevant to human disease. How can we leverage this developmental view of the mouse? I mean, we're talking about mice. How do we actually try to understand how this is reflected in its relevance to these tumors that I was talking about, medulloblastoma? So we had actually some medulloblastoma, and the nice thing I want to say about microarrays is that this community, along with the SNP community, has been unique in making freely available the data to its community. For those of us who have MDs or will have MDs, let us be aware that still today, most clinical research papers do not publish the primary data from which the conclusions are made. Still do not. and therefore you are unable to independently verify these results or come up with better results or with improved results. And that's why it was really incredibly important, and I'm sorry I didn't emphasize this before, that the early practitioners of microarrays, such as Todd Golub and Pat Brown, published all the data right away after publication. In fact, I looked at a half year ago at Todd's original paper in 1999 on AML versus ALL, and I thought there were 700. I think there were 700 citations of just that one paper, and it would have not been the same had they not published data, because there was a lot of secondary analyses and a lot of problems found in the paper and positive sides of the paper. It sets up a whole scientific process in motion that you don't have otherwise. So we have this data on a variety of different human medulloblastomas that have been published a couple years ago. And then we could go to a database called homology. And by the way, your next problem set, not the one I'm describing to you, but your next problem set will be a treasure hunt, where we're going to make you hop across all the different databases that you should know about as basic, not basic biologists, but basic consumers of the very simple kinds of information around about SNPs and the microarrays. But we'll get to that treasure hunt later. Suffice it to say that there is a database called homology, which allows you to map the genes in humans to those that are present in mice. And it turns out that there were present on those two microarrays, they're on the order of about 3,000 genes that are homologous from mice to men. And so what we did then was to do a principled component analysis of these data sets. Now, hands up those of you who really understand principled component analysis. Okay? Two of you. So those who don't, let me try to give you the simple version of it. Principled component analysis essentially tries to refactor the data so that you're looking at these new variables that are linear sums of the original variables, in this case the genes. So these principal components are linear versions of these original measurements that capture in successive components the major amount of variation of the data set. So the first principal components is this linear combination that gets the largest amount of the variation in the data set. The second principal components are orthogonal, essentially in right angles to that first principal component, and get the second most amount of variance, and so on. And in fact, you have as many principal components as you have original variables. But the nice thing is that, heuristically speaking, by the time you've gone to about 2 or 3 or 4 principal components, you've typically captured on the order of 70 to 80 to 90% of all the variance in the data set. In other words, by doing these linear combinations of orthogonal vectors, you're able to capture most of the variance. And the nice thing about that is for data sets such as gene expression, where you have a lot of highly correlated behavior. In other words, the genes are really not, there's large numbers of genes that are really not that independent of one another. This allows you to reduce the number of variables that you have to look at an analysis from a very large number, tens of thousands, to just a handful, the first few principal components that allow you to capture the majority of variation. And that's, by the way, the intuition that some of this face recognition software that you may have heard about from Homeland Security has been inspired by essentially taking the eigenface, where they look at the principal components that make up the features of the face and reduce the complexity to these principal components of the face, so that not only can you generate an eigenface, this generic face, but you can also map people to which faces they're most like. So it allows you a little bit of play and slop between different angles of and different ways that you might look in a different day, because it's still capturing the major variance of your face. So what we did is the following. Not only was there day 5 of the mouse, but we actually captured multiple time points on the order, I can't remember now exactly, maybe 15 time points. And it's a publicly available data set if you're interested. 15 time points going from embryonic days to postnatal days, day 60, I believe, of the mouse. And then when we did the principal components of the genes expression pattern across this time series, we saw the following when we just plotted the genes by their position in this new coordinate system, the first and second principal components. And I've colored them by these, whether they fell into this section or that half section of the egg, so to speak. And why do I color it so? Because if you actually look at when these genes had their maximal period, their maximal time of expression, the ones in this component, in this area of the two components, had an early peak, and the genes in this component had a late peak. So the early principal component and the late set of principal components. In plain English, by just doing this simple decomposition, this simple factoring into the two principal components, we've taken that time series and replotted the genes, and now we have a set of genes that are clearly separating themselves out from another set of genes, and they're characterized by being early versus late peaking. Now, so what? We then took the human homologs of those genes, and we asked in the medulloblastomas which genes compared to non-medulloblastomas were upregulated and downregulated in the human tumors. Please.
The previous slide, or this slide, the x-axis and y-axis, what's the
There's the first and second principal component.
Yeah, but the numbers like 0, 1, 2, 3, and...
It's I can't. I don't think it's zero based. I think 0 is somewhere around here.
But so it's just. I know. So what does that mean? It's according to.
It's according to the coefficients. If you position that gene according to its position along those two principal components. So each one of these is a single gene. So then what we do is we plotted those up and down regulated genes by whether they fell into, we just took those up and down regulated genes without knowing anything more, and we say where do they fall into this late and early decomposition based on the principal component analysis. And the simple answer was essentially 90% of the genes that were upregulated fell into the early period of expression. And similarly, around 90% of the genes that were downregulated, in humans, mind you, fell into the later phase of development of the mouse. Go ahead.
I have a question. The way you're presenting the data, it doesn't make apparent why you need the second component.
That's a very insightful question. And the answer is that for what I'm describing, it does not. Because the separation that you're seeing is just in one dimension, and you're absolutely right, it doesn't add anything. Turned out there were actually different processes that I'm not describing here that actually were separated out by going into the second principal component. But I think you're showing that you're understanding what the hell I'm talking about by saying, for this distinction, early and late, all you need is the first principal component. You're absolutely right. So I just want to, those of you who've taken basic pathology in medical school will hear the following, I don't know if I have a pointer to a slide, but it's been known by Kohane and others from the turn of the century have speculated that there's something about the embryonic program of development that is recapitulated by tumors. That tumors are similar, not the same thing, are similar to essentially a poorly controlled embryonic development. Because when you look under the microscope, a lot of the tissues in tumors are not as well differentiated as the tissue from which they came from. liver cancer, if liver cancer looked exactly like liver tissue, it wouldn't be liver cancer, it would be liver. And so they're actually more primitive versions. And this actually adds support to this. In fact, just to check on this, we did the following experiment, which I don't show here. So what we've done here, just to place this in the appropriate perspective, is take neural tissue from human tumors, and projected it against the principal components of mouse development. And what we see is this nice separation between upregulated genes and the early phase. Now, maybe all we're showing are general markers of hyperproliferation and not something specific to the system. So what we did is we took a similar pair, namely lung cancer and lung development. And lung cancer and lung development showed essentially the same relationship. But then when you took medulloblastomas against the lung development background, the separation was no longer as good. And when you took the lung cancer against the neural development, it was also not as good. So what it tells us is something much more nuanced than simply embryogenesis recapitulates generically oncogenesis, or the other way around, I should say. It says that there is part of the differentiation program of that particular tissue, which is recapitulated by the tumor. And that's already kind of interesting because it gives us some insights in development. But it gets more interesting when you start thinking, well, I know a lot about different stages of development. Can I actually start pulling out my understanding of the mechanisms operating at different stages of development to understand what's different about these different cancers? So this is just showing you in the time series what the principal components were showing you before. And in red and green, we're distinguishing the genes that are up and down regulated. And you see they have, in the human tumors, they have different time courses in the mouse data versus in the different courses in the mouse data, depending on whether they were upregulated or downregulated in the human tumors. And as I say here, this is a slide I wanted to. Lobstein and Kohane were among the first to theorize similarities among human embryogenesis and the biology of cancer cells. And it's actually not until now that we've gotten really much more objective evidence that this is the case. And the brain tumor classification system that is used, and that was divided by Bailey and Cushing in 1926, from which modern taxonomies derives, emphasize this. But they're very crude taxonomies and descriptive taxonomies. And here we're providing perhaps, and is that, I'm saying perhaps, a much finer classification now. Is that true? Well, let's turn around the principal components. Let's look at each tumor as a function of the genes that are upregulated. Let's look at the position of these genes, of the tumors now, not the genes, by the position in the principal component analyses. And to make a long story short, we were able to dissect out different days of development. So these are different mouse samples, and projecting onto different cancers, human cancers, onto the same principal component analysis of these mice. And this is very interesting in a number of different ways. One is we're separating out different days of development into this principal component space, but also separating in that same space the tumors. Now that's fundamentally interesting because it's telling us, and the reason why am I bringing this up in a genomic medicine class? Because if one of the fundamentals of modern medicine is taxonomization of disease. And here I'm providing a very, very quantitative measure of differentiation of these different tumors based on their position in the space of development from a data set that you could never get from humans, but from mouse. And this is truly exciting because I don't have time to, I just don't have a slide to show you because we just submitted this, and sorry, it just got accepted to Genes in Development, which is an important journal in this space. We can actually separate the metastatic from the non-metastatic medulloblastomas in the same way. They separate in the different space. And so what does this tell us more generically? Principal component analysis is not a commonly used tool for clinicians, obviously. And yet here we have a tool that is creating a much better taxonomy of disease with predictive power previously not available to us. And and allows us to understand, because we're going to say what are the genes, because remember I said the principal components are a linear component, are linear combinations of genes. We can say why is this gene in this position? Which genes have the major weights which make it responsible for that tumor being in that position? Might give us some insights into the underlying tumor mechanism. And so here we have a fine-grained nosology which was not previously available to us. And I think it's therefore not a leap to think that in similar kinds of processes, the computational approach to these data sets will allow us to recast our very, very squishy current taxonomies. I think I told you in the first lecture, with a few exceptions, and the most notable exceptions being microbiology, most of our disease classifications are not mechanism-based. In microbiology, the disease classification is mechanism-based. It's the organism that's infecting you. That's the name of the disease. But most other diseases like lupus, so probably a mixed bag of inflammatory diseases that we don't know, polycystic ovary diseases, we're describing symptoms, not pathophysiology. And I think this is allowing us to get much, much closer and therefore a much more robust and finely grained understanding of that. So my interim summary here is that this computing allows us to identify some pathways and provides, more importantly, a natural classification of disease with further insight into mechanism. Apparently, I don't know how to hold the microphone. Go ahead.
Did you try predefining vectors that you assume could evolve from meaning line against the optimized combination of genes that you'd expect to be a developmental program or something to that effect?
So Jose is asking a very interesting question, which the short answer is no. But I think it's a very good question, but I wouldn't know exactly where to start. But surely there is a thoughtful way to respond to it. Could you take linear combinations of genes, and I wouldn't know exactly which linear combination of genes, but maybe just using those as a starting point, perhaps. Yeah. Genes that have been implicated in various stages of development and using those. And I think that's a good way to go. My understanding of developmental biology is unfortunately it's very sparse knowledge at different stages, so it'd be hard to know which genes are generally informative. Nonetheless, I'm a big fan of the approach that you're suggesting, which is a knowledge-based approach. What we're doing here was clearly knowledge-free. We were just taking the data and just saying what explains the maximal variance and going from there. But if we could if we could see that process with a set of genes that we knew were implicated, I bet we'd have much better resolution than we have currently. So I think it's a very good thought, but we have not done that. Which brings me now to another aspect of computing. I've shown you guys this slide? Doesn't look familiar? Good. So here's the fantasy that most deans of medical schools and heads of hospitals have. And again, this is not a very technical point. It's more of a sociological point, but will end up being technical. Because it's going to tell you about a whole other area of biocomputing that I think is ripe for the picking, but is also a rate-limiting step. So they all have the fantasy. Now that they've seen things like, oh yes, I can predict different outcomes of different tumors, let's say, I want to be able to develop a set of targets that a drug company is going to pay me a lot for. So the fantasy is that a nice doctor talks to the nice patient, gets consent in a nice hospital, they obtain a family history, they bank the tissue obtained, they do some genomic analyses, they do clinical annotation, do some fancy mumbo-jumbo bioinformatics, and lo and behold, they're going to have the target that the drug company is going to pay a lot of money for. That indeed is, for those of you who are more entrepreneurially oriented, that's the fantasy that's launched many venture capital funded ships, like the Decode Project and others. And the problem is the following. It's that this central piece, the phenotypic annotation, ends up being the hardest part. Understanding what really happened to the patient. Did the patient really have a tumor? How old were they when they had the tumor? What drugs were they responsive to? That is the hard part. And any of you who have ever looked at a clinical chart can understand why that is. There's just very little useful machinable data in the clinical chart. Because in fact, the real fantasy is the following. In what I call Ratschwitz at the Medical College of Wisconsin, what they do is they take rats, consomic rats, where they have systematically substituted one chromosome for another, so which gives them very big efficiencies of identifying linkage with a different trait. What they do is they take these different consomic rats, different strains with well understood genotypes, and they expose them to very well characterized environmental exposures, such as hypoxia. They put them in the oxygen equivalent on top of a mountain for several weeks, or they give them a high salt diet or high fat diet, or they volume deplete them. And after that wonderful experience, they then physically deconstruct them and, for instance, rip out their heart and put their hearts on these prep machines where they can look at the contractility curves of the heart in a perfused prep. And that's great because now you have a well-defined phenotypic environmental characterization. You know the genotype and you have the expression. And by the way, let me say and point you to the following site, pga.mcw.edu. They have all that data online, the phenotypes, the genotypes, and the expression. And they do it for cardiac phenotypes, kidney phenotypes, lots of different phenotypes in different parts of medicine. And so this is the fantasy. But of course, patients would probably object if we subjected them to this kind of treatment. So this high-throughput phenotype fantasy just is unlikely to happen anytime soon. So we took a very different approach when we were dealt with this problem. In other words, how to get sufficient numbers of samples in the right amount. And you should know that there are companies like Ardeas, which have been given millions of dollars of venture capital unsuccessfully to try to solve this problem, to get enough tissues in the right amount. So the National Cancer Institute put out a request for applications saying, listen, there's thousands of tissues available throughout the pathology repositories of our country that we want to be able to do genomic studies. How do you go about that? So inspired by Napster and Nutella, and for those of you who don't know what those of you who are not aficionados of file sharing, Napster is a file sharing service where the directories are centralized, but the data themselves are decentralized in multiple directories. Nutella, in order to avoid getting anybody sued, is fully decentralized. The directories are decentralized, and the data is decentralized. And what we built, the Shared Pathology Informatics Network, SPIN, that's funded by about $7 million from the NCI, is exactly the same thing. And what we did is to take advantage of the twin obsession compulsions of pathologists. Pathologists are probably the best first taxonomists of the medical profession. They actually categorize organs and specimens better than anybody else. Second, they're obsessive compulsive collectors of tissue, and they keep tissue around for years and years. And so we thought that if we could take advantage of those twin compulsions, we could actually be successful. So at a bird's eye view, what we did was the following, which was to create a network whereby a user with a web browser could send a query to this query composer, which would then send out a query to this nebulous thing called SPIN network, which would, using this peer-to-peer technology, respond which samples were out there. More specifically, each tissue bank or institution would have its own node on the network, on the internet, and we'd provide them these open source free tools which allow them to extract from these pathology databases, because there's a textual report with each pathology sample, both the anonymized, because we have this anonymization program, the anonymized textual report, and code a few data elements to search, so that when you put in the query percolates in a fully distributed way. There's no center to this network. This query percolates throughout the system, all the nodes respond eventually, if they can, and you get back the sum of all specimens involved. And those of you, even in the medical profession, probably don't appreciate the following point. The reason we created a peer-to-peer system is that pathology tissue banks are jealously guarded by the pathologists and by the surgeons who built them. They just don't want to share data. And it's essentially impossible today to know, even across the street in the Brigham, what samples they have. And by allowing them to actually fully control their own node and control what they expose to the outside, we're able to overcome the sociological obstacles for data sharing so that we can actually do this. So, for instance, we've done this now. This is actually an out-of-date slide, not only across Harvard and UCLA, but now we've gone live with Pittsburgh and a few other sites. So that today, if you're a registered user, you can actually send out a query to all those institutions that you see on the right. And the query then percolates across the country. And this query takes on the order of 10 seconds on a bad day to execute. So for instance, we want to know how many distinct specimens could we have of renal transplant patients. And that's typically hard to come by. And here was the response. So this is out of our 7 nodes. This is a query I did back in October of last year. Out of the 7 nodes that we had up at that time, with only 2 nodes responding, we already had 20,000 specimens identified, with an age distribution shown as here. By the way, I'm aghast at the fact that apparently 34 specimens of patients ages 90 to 99. I wish it was true that that was an error, but I looked into it and it's not. So some weirdos are actually transplanting some very old patients. Would not happen in England. In any case, here's the age distribution, and if you click on the full text and you have the appropriate privileges, you can actually see the full anonymized text that will report on that patient. And so I make this point for a number of reasons. First, we're successfully mining this obsessive compulsive feature of pathologists. Second, I hinted to you that some of these outcomes analyses using functional genomic measures, such as the lymphoma study, are essentially not well reproduced in different studies. Whatever the reason, and they are many, a lot of the ills in analysis are excused by large numbers. So if we went from having 100 patients to having 20,000 patients, I assure you that the differences in different machine learning techniques would matter very little. And so this is how we're beginning to approach how do we harness the phenotypes that are out there that are available, that are not available otherwise, through this a completely different kind of bioinformatics. This is more infrastructure kind of bioinformatics. But it's the rate-limiting step. We can work on dozens of samples until we're blue in the face. We really won't know how much we're overfitting our data until we have large samples.
But is there any sense saying that you can request those samples and then do studies?
Yeah, I wish it was that simple. So that shows you're awake. The next question is, and how do I get the samples? So I wish it was true that once you identify a sample, you just click the box, put your credit card, and you get FedEx the tissue sample. So what happens at that point is that you start a dialogue with the investigator and the institutional review board of that tissue bank. So that's the bad news. The good news is we've solved one part of the problem, is figuring out where the specimens are to find out if your study is feasible. And people are already beginning to use this tool right now to write grants, because they can say, I know that there's this many specimens out, and they don't necessarily get access to the tissue for that, but they get in principle agreement from the collaborators that if they get funded, they'll do this as a collaboration. But you're absolutely right to do it. So this is just another example how we're using computation to overcome the obstacles to doing genomic medicine. Let me, I think, give you one last case history, and then we'll go to the problem set. So let's talk about how we can use computing again to have a different understanding, and how we use computing allows us to have a different understanding of, for instance, gene regulation. So this is time on the x-axis, and this is some arbitrary value of expression. Are these two time courses the same? Are those two genes developed by the same process? Yes or no? You can make an argument either way, right? Maybe they're the same. How about this one? Maybe they're the same, maybe not. I told you the stuff was pretty noisy. Maybe that's just noise. A priori, who knows? Now, does order matter? In our analysis, does time matter? Yes. Good. Because it should be true that the likelihood of me being here now is informed much more by where I was 5 minutes ago than where I was a day ago, right? That's a fundamental Markovian property of Zach and most physical processes. Now, does the correlation coefficient at all address that Markovian process? In other words, if you take two stocks and shuffle the timing, let's say two stocks, Apple and Microsoft, and let's say for the sake of argument that they are highly correlated, they have a high correlation coefficient. If you permute the days, but you maintain the same pair of stock values for that day together, if you shuffle the days, is the correlation coefficient the same or different? Same. So correlation coefficient does not, in fact, capture the effects of time. Now, as I hope you're aware from our previous discussions, a lot of the clustering that's been done, even on time series data, is done on the basis of correlation coefficients. People in those dendrograms bring together genes that share the same share the same expression pattern as measured by the correlation coefficient. So what consequences does that have? Let's go back to a very old study. This is Ayer's study back in 1999, a classical Jewish study where they snipped off they took fibroblasts from foreskins and they measured the gene expression pattern over time of the transcriptome of these foreskins after being exposed to serum. So you see here every column is 0 hour, 15 minutes, and so on to 24 hours of each gene. And then they did this act of creativity, which is they draw lines next to the dendrogram and then cut out the blocks, each line, and say, oh, look, there is a bunch of these genes, and they seem to have, for instance, coagulation hemostasis. There's a tissue factor, pathway inhibitor, and so on. These are coagulation involved. These are cell cycle proliferation. This is inflammation. Oh, look, these are angiogenesis involved genes. These are cytoskeletal. And they all seem to be clustered together. And it's, again, I think I use this metaphor all too often, but it's so true. This is very much like the dog at the opera. It's a miracle it can sing at all, and you don't criticize how well it's singing. Because what they've done is this act of creativity. They've actually looked at the data of genes, and they draw these lines and say, these belong to a chunk. Now, there are other lines they could have drawn, and in fact, if you look closely at their original paper, there's large numbers of genes that they do not draw those lines next to. They don't have anything to say. So what's really going on here? So again, these clusters are defined by correlation coefficient. So what if we tried to take advantage of the Markovian property of gene expression, like any physical process, namely that in a time series, the recent past informs you more than the distant past. So you can approximate the conditional probability of this value upon the recent past rather than the whole time series. That is, the basic assumption is that T0 is independent of the remote past given the recent past. And how many steps back you look is what you well know as the Markov order. So if Markov order is 2, that means you look back 2 time slices. If Markov order is 1, you look back 1 time slices. If time ordering does not matter, just like the correlation coefficient, then your Markov order is 0. The recent past actually doesn't inform you at all. So, now let's look instead at clustering as a statistical model selection problem. So rather than looking for highly correlated pairs, we're going to consider each of the time series of these genes as being examples of a process. And the process is going to be represented, therefore, by one or more genes which are driving that process. And the question is, for any given pair of genes, do they belong to the same process? And essentially, with a very simple Markovian analysis, which is just a pain in the butt to implement, you apply Bayes' theorem and just say, is it more likely that these two gene signatures were generated by the same process or by a different process? If they are generated by the same process, if the model that they were generated by the same process has a higher probability than the one that they have a different process, then you cluster them together, just as you would in a regular dendrogram. Of course, the threshold you pick, the probabilistic threshold that you pick, is going to determine exactly which branches are brought together, which gene traces are brought together, but nonetheless, you now have a strong probabilistically based and Markovian-based reason for bringing together two gene expression signatures solely based on the marginal likelihood of these two gene expression patterns being generated by one process versus the other. And you can thereby eliminate them appropriately. Since I'm running short of time, let me just tell you that the Bayesians have a number of nice tricks in their armamentarium, which essentially depend on the following observation, that if we use the same data for all the models, and that if we assume all models are initially equally likely, then you can do that very nice equivalence shown at the bottom, such that you only have to calculate the marginal likelihood rather than the absolute probability in calculating which model is the most likely, and therefore it simplifies a lot this calculation. And incidentally, this program that we called Caged is available publicly for if you look at time series. But what does it do? So if we took the same time series that you looked at with the expression data from the foreskin through this same Markovian clustering program, we only found 4 clusters, 2 small clusters and 2 large clusters. So what's with that? The two small clusters were one a cytokine cluster and then an apoptosis cluster. And the large 2 clusters had a bunch of different genes in them, and they contained essentially all the other clusters that had been obtained in the original analysis. So what, you could say at this point, well, Zach, you guys didn't do a very good job, because despite all this convincing talk about the Markovian nature of gene expression, they were able to really pull apart a lot of processes, and you were not. So how do we know that we did a better job or not? Well, it turns out that back when they did their original experiment, Unigene, which is a database that Alberto will tell you about, was an earlier stage. And what do I mean by that? Every time GenBank grows by a certain amount, by which I mean more sequences are deposited into the human genome database, there's a periodic reassembly of the putative genes based on the better and better populated jigsaw puzzle of GenBank sequences. And sometimes it means that two things that were thought to be different genes, when you have enough bridging sequences, ends up being the same gene, and a gene that was previously thought to be the same, with enough distinguishing sequences, ends up being split into 2. So these unigene builds are different. And consequently, when we look back in 1999, 238 out of 517 genes were unknown. And we relabeled the genes according to the current state of the art, at which point only 20 were left unknown. And there were 19 genes that were present twice in the data set. And the original clustering puts 4 of these in completely different clusters, whereas we only did this once. If we put our Markov order back to 0, that means we ignore the past, we get exactly the same misclassification of these identical genes. So the conclusion here is that the temporal ordering does matter, and doing just a simplistic correlational analysis will not cut the mustard when you're really trying to dissect some of the processes. And using a statistical measure of clumpiness of these clusters rather than a looks right test, which had been the standard for most of these papers from 1999 to 2003, probably is also not a safe thing to do. So it turns out those two traces I showed you before were in fact the same gene, although it was not known to be the case when it was first published, and these two traces were put in two different clusters. And likewise, these two traces also ended up being the same gene, and they too were put into two different clusters. All right, let me wrap up by talking about another aspect of genomic medicine. Those of you who have done clinical research know the following is true, which is most clinical trials are censored, by which I mean either some patients drop out of the trial before it's completed, or the patient has died by the end of the trial, if mortality is your endpoint. So it's unclear whether, for instance, a patient who just died right after the end of the trial should have been included or not. And the big picture is that by having a particular cutoff point on the study, you're not allowing yourself to see the full evolution of the patient's history, and therefore, depending on what the nature of the study, you may actually have a very strong bias. And it's been shown in clinical research again and again that if this censoring is not attended to, you'll have a misinterpretation of the clinical trials. So that's well known in the clinical research arena. And the question is it equally true in genomics as applied to clinical research? Well, let's understand what most of the studies published actually do. They often do the following. They take a set of gene expression patterns. They do some clustering operation, such as what Alizadeh did originally in their paper. And they say, oh, look, there's two or more natural clusters just based on the gene expression profile. And then they ask themselves, after they found these expression profiles, is there anything different about these patient populations? And they rummage around, and in this case, they found a wonderfully different mortality between the two groups of patients as defined by the expression pattern. So let's review that. You first look for a difference in expression pattern, and then you say, what makes these patients different? And it makes for a very impressive publication, because you're not looking at the patient, just saying, I'm looking at gene expression pattern, and then when I open my eyes and allow myself to see what's different in the patient in these two groups, I'm finding something that's really clinically different. Now, what's the problem with that? The problem with that is that you may be looking at the wrong thing, for example. With 10,000 genes to look at, there might be a cluster of genes that, for instance, let me just actually, before I come up with a fanciful example, it might be that the reason that these patients have different expression profiles is because they weigh a different amount. One group is fatter than the other, because one group is sicker than the other, except it was not picked up clinically, but one is on the way, is going down the tubes, and one is not. And what we're really seeing here is not something intrinsic about the tumor, but something intrinsic about the weight. And so what we're getting at is a very indirect measure of mortality, and it may be very much unrelated. Can we do better by going directly from the gene expression pattern to the clinical phenotype of interest, rather than going through this two-step process where you first cluster the genes without any knowledge of the phenotype and then see how the phenotype is different? Specifically, can we directly find genes or linear combination of genes that are highly correlated with survival times. For example, gene A plus 0.5 times gene B plus 2 times gene C equals some probability of survival time. Can we do that? And can we do that in the context of the kind of censoring that I described? So in summary, how can we use the survival times directly to find good predictors? So here's a fundamental data problem. We have gene expression data, we have phenotypic data, and we want to find out how one predicts the other. Now, previously, if I asked you that question, you'd say, oh, Zach, let's just use linear regression or logistic regression, for instance. Turns out, with 20,000 variables, it breaks down. Just does not work. And so a nice answer to that is something called partial least squares. Now, what is partial least squares? Well, let me give you the intuitive feeling for it. I explained to you what principal components were. Principal components being these components that capture a large component of the variance, and the subsequent components are orthogonal to each other. This is like principal components, but as they relate to a specific outcome variable, like survival. So these are principal components that are correlated essentially with an outcome variable. So that's interesting, but the problem with partial least squares is that it does not actually allow you to use censored data. It assumes that the data is complete throughout, and that's going to immediately bias your data. So how do we take advantage of censoring? Now, in classical clinical research in medicine, what you use is something called the Cox model, which essentially is a probabilistic model that says, what is the hazard of dying for this population of patients, and basically imputes for the missing data, fundamentally. And for a small number of variables or genes, you actually can use a Cox model to actually figure out, essentially, what would have happened if you'd had complete data. But the Cox model, just like regression, does not work well for tens of thousands of variables. So what are we going to do? How do we get to use partial least squares? To make a long story short, and I guess I'll post a paper to my course's website. What you do is you actually model the patient's data using a Poisson regression, so that you actually transform this data set into one with missing data, from one with missing data to one with complete data, based on this Poisson regression. But the problem is now we're going to create essentially many more data points, essentially. And just to give you an intuition behind it, because I don't have time to go into a full description, is this will allow us to estimate the following problem. So we know, let's say that a patient lasted until this point and dropped out of a study. Where might they have died? What can we assume based on this prior data? So by using a Poisson model, we can enter all these new data points, and so we can actually figure out precisely, well, probabilistically, I should say, what the values were at the various time points, and completely in this probabilistic sense. So now we can apply partial least squares. So what does that mean pragmatically? So Bhattacharjee, how do you pronounce that? Bhattacharjee, and et al., basically working with Todd Golub, did a study of lung cancer, which had, in fact, censored data. And each patient had a survival time, and it was marked whether the data was censored or not. And the question was, could they identify different outcomes? And the short answer is they were able to identify, for instance, metastatic versus non-metastatic, but there were a lot of other outcomes that they could not distinguish from one another using these 125 samples. So we asked ourselves the question, if we took into account, using partial least squares on top of this Poisson model, could we actually tease apart in a reliable fashion some mu phenotypes? And the short answer is, we were actually, these are uncorrected p-values. Whoops. These are uncorrected p-values, but 10 to the minus 7, even if you do correction for multiple hypothesis testing, you can see that they're actually going to be highly significant. We were able to obtain components that were highly predictive from the partial least squares analysis, highly predictive of clinical outcome, where previously you could not. And to make that very clear, these were two groups of patients that previously were not distinguishable, but by using this method, we were able to distinguish them with a p-value of this amount and provide a very sharp distinction between these two groups of patients that was actually not resolvable using the standard analysis that done previously. And the reason is, because I just want to bring this home to you, because we were able to go directly from gene expression to the outcome of interest. We didn't ask ourselves, let's cluster the patient data and then see how the patients are different after the fact. We said, I know the different characteristics of the patient, in this case mortality. What combination of genes best predicts that difference? And by looking at that direct signal rather than indirect signal, with all the complications of having to deal with censored data, we're able to separate out very, very well these subgroups of patients. So again, showing you how by using a little bit more sophisticated computation, we're able to identify subgroups of such patients. So this brings me to the problem set, and I'm going to spend a little bit of time to make sure you all understand the problem set. So I think it's very important for you to get your hands dirty with the data. So there is a data set that's available at the following URL. And if you just Google Kunkel, Kohane, Hazlitt, you'll probably find it, but that's the full URL. And I'll send out to the group the URL through our group email. And that's a data set from a paper that we published, I think, a year or two ago about Duchenne's muscular dystrophy. Duchenne's muscular dystrophy is a degenerative disease of the muscles. It's the major cause of what Jerry Lewis raises money for. And we have data on the muscles of patients who have this disease and people who don't have that disease. And what I want you to look at is that comparison, and I want you to identify those genes that are differentially expressed. Now, if you want to work in pairs, that's fine, but no more than pairs of you. And I'd like you to use at least two different tests. One is SAM, Significance Analysis of Microarrays. The other is the t-test, which I hope most of you know. There's many toolkits that you could use to do this comparison. Let me recommend to you the MEV, the Multiple Experiment Viewer at Tiger. Tiger is the Institute for Genomic Research, and at tiger.org. So if you Google MEV at Tiger, it does many more things than just these two tests, but I recommend it to you for doing these two tests. So given these data, I want you to answer the following question. What is the difference between the top ranked 50 genes, differentially expressed, both up and down by the two methods? And why are the lists of genes different? I'd like you to use another program called MapFinder to classify these genes by their function. And so the output that I'm expecting from you is two sets of differentially expressed genes based on these two different methodologies, your explanation why these sets of genes are different, and a classification of these two sets of genes by functional annotation. This is a very simple exercise, but I think that it's important because if I've learned anything about this area, it's just doing it by yourself a few times opens your eyes up to how bad the data is and what the problems are in analyzing it. And if you're not comfortable in downloading tools, then partner with someone in the group who is. All right.
Can you post this on the website?
I'll put, Long will post this on the website, and I will send an email out of this URL. Any other questions? That's an excellent question. It's now February 24th. How about when's March 15th? Can you look up what day of the week that is?
It's a Monday.
So March 16th. And please let me know if you're having problems. The guy who actually built MEV is a friend of mine at Tiger, so I can kick his butt if it doesn't work for you at a particular time. All right.